So Dr. Xiangrui Li is the Ohio State University CCBBI facility specialist, aka the jack of all trades for our center. He received his PhD in neuroscience from USTC China and has been at OSU for over 9 years. So please join me in welcoming Dr. Xiangrui Li, and I'll let you take it away. Thank you.
Thank you, Alison. And thanks everyone for joining today's session. So today I'm going to talk something about the NIfTI analysis using Matlab. So as we all know, NIfTI is a very popular image format. So almost every package, I think only exception probably is Brain Voyager, adopt the NIfTI format for their as their image format. So for most analysis, our first step to do the imaging analysis is to convert Dicom to NIfTI file, because almost all scanners are designed for medical purpose, and they always save the images as a Dicom format. So because Dicom format, although it's universal, but it's kind of too complicated for our purpose. So that's why in the scientific area, people prefer the simpler NIfTI format. So far, I think the popular SPM, FSL, AFNI, they all use NIfTI format. So as SPM is Matlab based, it has its own Dicom to NIfTI converter. Should works pretty good for Siemens data. The one thing people may not like is SPM, or until I think I have no idea for the latest version, until at least to the SPM 12, it still save one file per volume. If we have 500 volumes for a run, it will save 500 files, NIfTI files. So that's kind of a doesn't look good, but kind of a speed up the file reading a little bit. And the other two popular Dicom to NIfTI converter is the Dicom to NII. Actually, the current one is DCM to NIIX by Chris Roden at the University of South Carolina. That's a compiled version, runs pretty fast. The other one is I developed in Matlab. It's called, the name pretty similar. There's an extra I. I always put a .m to distinguish from that one. So inside my package, I also have, there's also the NIfTI view. That's a very useful visualization tool we are going to use. I'm going to show you today how to use it. It's pretty convenient if you use Matlab. So image pre-processing, these are the steps. That's not something I am going to cover today. Maybe if there's a need, well, multiple people in a students group or other people, we can together do a session to cover the image processing. So today, the example I'm going to show is the image, mostly in the image after pre-processing. So the pre-processing step, they're pretty much similar. They are not exactly the same, but pretty much similar among the major analysis packages. So anyway, in either in any of the major analysis packages, image visualization is very important. You don't have to visualize every single subject, every single run, but at least for the testing run for a participant, at the very beginning when we develop the pipeline, it's very important to visualize each step, make sure everything works fine before we submit to a scrape or something to do it overnight. we don't want to do it 3 days. Turns out everything's wrong. So visualization is a very important tool to avoid some unexpected problem. So most analysis packages like SPM, FSL, AFNI, they all have their own visualization tool. Free Surfer, like Brain Voyager visualization tool is pretty good, but the Brain Voyager, I think, is the only commercial software. Is pretty expensive. So I think the user population is relatively small. And here I need to emphasize the FSLIs in the later FSL. That seems very powerful. Actually, the motivation I developed the NIfTI view in Matlab by myself is because the FS view in the older FSL version, FSL is really not nice. I think the major reason is because they try to take care of the the obsolete analyze format. So the support for the NIfTI was really bad. So for my own purpose, I can't do a lot of things. Then that's one of the major reason I tried to develop the visualization tool in Matlab. Also in the Dicom to NIfTI tool, this one, there's a MR cron that probably is it's a very tiny, compact tool. It's very convenient. If you run Linux, it's very nice, easy to download, easy to use. And nowadays, some people, I guess some people we have started the HCP pipeline. They have a tool called a Workbench, and it provides the visualization to both traditional volume-based space and surface-based space. So later, I also make my tool compatible to the HCP surface. So we can take a look how beautiful it is. So because my tool advantage is I can claim it supports virtually any NIfTI format. Version 1, version 2, most people don't use it except HCP people. And volume, surface, any data type, NIfTI support. So not exactly. Some NIfTI support some weird format, like the double, that's not supported because it's not supported by Matlab. So here's some basic thing about NIfTI. And NIfTI file is very simple compared to the popular Dicom. So it has the header and image. That's the major two port. Then in the middle, there's an extension port, but most NIfTI file don't have it. So the NIfTI header has a fixed length, a different version 1, version 2 length are different, but it's fixed. The important ones we should pay attention. We don't need to understand all those things. Some of them are totally doesn't make any sense at all. So the important one we need to pay attention is the dimension. That tells what's the dimension, image size in each dimension, X, Y, Z, 3D, and 4D typically is time. Typically those the 4 numbers. And the voxel size, what's the X, Y, Z in millimeter, and what kind of data type. Typically from scanner is kind of a 16-bit integer, but whenever we go to process, that's not enough precision. We will use a single precision float. In Matlab, default is double precision, but single precision reduce the size by half. So that's why most packages use a single precision. The other important information in the NIfTI header is the so-called form, NIfTI form. And each form is corresponding to a transformation matrix. The transformation matrix we'll mention later. That's a very important feature in the NIfTI file. That and compared to its old preset, I think that before we use NIfTI, people use Analyze. Compared to Analyze, this is new, mainly the new stuff. So and this solve a lot of confusion existing in Analyze format. So extension is a pretty powerful part of NIfTI format. Unfortunately, there's no consensus to use it. So that's why most software packages don't use it. HCP is the mainly one, major one to use it. And because it will save a lot of information and you can save any information you want. You can define your format, but the problem is that I don't know your format, so it makes other people hard to understand. But that's a flexible part of NIfTI. And image part is stores the data, image data. Typically, it's a 3-dimension, or for functional data is or diffusion data is 4-dimension. The NIfTI tool in my package, in the Dicom to NIfTI package, can read, create, and write NIfTI files in Matlab. We will go over that later. It's pretty convenient. So again, like previous 2 seminars, I'm not going to go to very detailed for the NIfTI, what does each parameter mean, header parameter means. I'm not going to that. That's very, if you are interested in that, it's very easy to do a Google search to understand those. So what I'm emphasizing here is while we all use image format, what is easy to go wrong for us when we do some special analysis, and how can we avoid those error in our analysis? That I think that's what I'm going to emphasize. So as I mentioned earlier, the NIfTI forms, that's the transformation matrix, is the most powerful tool, but it's also very confusing parameter for most users. For example, if we do the region of interest analysis, we need to pay special attention to what our ROI file is for. What's the form for our ROI file? It's typically it's MNI, but it could be other thing. So that's we need to pay close attention. If it's not MNI, we may need to do a conversion to make into an MNI space. Or not necessarily MNI, maybe our original scanner space. That's another, maybe that's a better option for a specific analysis. So for this most important feature, the matrix, the transformation matrix, here is, I take from one of our papers. So Dicom and NIfTI, they define direction differently. So for the NIfTI file, I'm not going to talk about Dicom. For the NIfTI format, the origin of the coordinate, it's not here, it's somewhere in the brain. It's the anterior commissure. And that's the tiny fiber connecting our 2 hemispheres. The middle of the tiny fiber is a point. We call we defined that as the origin of the coordinate. That means the XYZ coordinate of that point is 0, 0, 0, XYZ or 0. And then we define X, that's from left to right. That's the correct, positive direction. Y is posterior to anterior. And Z is from foot to head, or called inferior to superior direction. That's the positive direction. This kind of thing we should keep in mind. So the confusion part for the transformation matrix, there are mainly 2. is the transformation matrix diagonal major? If so not, typically we can reorientate the image into the diagonal major. So we will, what's diagonal major? I will show you later. The other one, this one is pretty, not very confusing because oftentimes like the major Dicom to NIfTI converter take care of this part. If it's not diagonal major, we make the diagonal major. The other one is the handedness. There's a left-handed storage or right-hand storage. Those are very confusing term. In the old atlas format, it's always equivalent to what we are now called left-hand storage. Although during that time, probably they don't have this terminology. And even if they had, it's not used because there's no another way. It's only one way. That's why in the analyzed format, you don't have a good way to tell left versus right. It's very easy to confuse left and right. But in NIfTI, that's completely solved by the transformation matrix. So when we get the transformation matrix, we use some important information in the Dicom. Here I list a couple of them, pretty 3 or 4. The most important one is the image orientation patient. That's the Dicom terminology. Those are 6 numbers, 1, 2, 3, 4, 5, 6. So basically it's a 2-unit vector like this. For example, for a slice, this is a Dicom image. And with this Dicom image, we have this parameter. This parameter has 6 numbers. And for a typical axial slices without rotation, it's typically something 1, 0, 0, 0, 1, 0. So those like a C, R, the first 3 define this structure, and the next 3 define this vector. With these 2 unit vectors, we can define the slice plane. And the second one is called image position patient. 3 numbers in unit of a millimeter. Like we call X1, Y1, Z1. That's the XYZ coordinate of this slice. One of its vertex is like this point. So once we have these 2 parameters, we uniquely define the position of this slice in 3D space. These 2 define the slice, and this one define the location. So that means this slice, where is in the space this slice belong to, these 2 parameters define that. The other one is how large each voxel is. That's X and Y, 2 dimensions. And there's a third one is how thick, if we have multiple slices, that's often the case. We stack each slice for perpendicular to the slice, to each slice. Then we have need a slice thickness. So we know exactly which slice is in 3D space. So with these parameters, it's easy to construct the NIfTI transformation matrix. Like R, the 3 R's, those are these 3 numbers. 3 C's are these 3 numbers. And VR, VC is these 2 numbers. One, this VR, this VR, this VC. And the third column is a slice direction. And this one, we could use slice thickness, but the other way is we use another slice location. That's typically we use a last slice location like this 3 number, but use the not for this slice, for the last slice. If this is the first slice, we use last slice. Then the difference between these 2 pairs divided by number of slices. That's those are this one, the first slice according to for the first slice. That's the image transformation matrix. So why is it important? Because once we have this matrix, basically, you can see it's a 0, 0, 1. We have only 12 numbers. It's a 4x4 matrix, but the last row is our constant. So why is it important? With this matrix, whenever you give me a voxel, like say, what's the first voxel, the coordinate for first voxel? Then, for example, if it's 0 base, will be 0, 0, 0. That's the first voxel. This part is the R. R times 0, 0, 0, 1 gives the XYZ of this XYZ coordinate of this voxel. So you can change IJK to any image index, then this give you the special location of that voxel. That's why the transformation matrix is so important. Let me switch to Matlab so to show you the header part. I'm going to load, just load any of the image file I have. That's the NIfTI tool I mentioned. It's just this load, this file name. So once we take a look, see there's a header image, but there's no extension. That's fine. And what I'm going to show you is the header. For the header, that's this part, that's the 12 numbers here. 1, 2, 3, 4, 1, 2, 3. This is one number. So as you can see, it's called S row X, S row Y, S row Z. Here, that's the NIfTI names. 1, 2, 3, 4, 12 numbers. Those are 12, this term number. That's the transformation matrix in the NIfTI header. So all the major packages use NIfTI format, but they deal with NIfTI format perfectly. So otherwise, we won't be able to use them. But when we do some customized NIfTI analysis, we need to read NIfTI into the Matlab or other package by ourselves, because oftentimes those packages won't cover those special analyses. In those cases, we will need something like Matlab. So like this is a load NIfTI, this is use NIfTI tool, load the file name, like I just did. And the next step we typically do is, for example, for the functional analysis, we will reassign the 4D data into 2D data. So I'm going to show you this, how to do this. It's very straightforward, very easy in Matlab. So as we can see, this image file has 88 by 78 by 44. That's XY 3D. And fourth dimension is 138. That's 138 time points. Typically, when we do analysis, we want the 2D image first dimension is time points. That's most time we, not always, but 19% of time, that's what we do. There are two ways. First way is, let's see, I just do a copy for the, it's just a copy the image to Y. If we see what's Y, that's exactly the same as the image. That's our original image. So to reshape the 4D data into 2D, so what we need to do is do this. I'm going to copy this. It's an error because when I copy from PowerPoint to Matlab, that character changed. So that's not, that's what we need to do. So if we check Y, it is in 138 by a huge number. So let me do this in separate step. First step is like this. Second step is something like this. So now the Y is same as image. Now what this does is we use the fourth dimension of NIfTI file, use that as the second dimension, and collapse all the first three dimensions into one dimension. So once we do this, we can see the Y is this number by the fourth dimension. That's the time points. Then the next one in Matlab is super easy. Transpose. That basically for the 2D matrix, it's swept the two dimensions. Then it becomes this dimension. That's typically what we need. So here's a time point. Here's a 3D we put into one dimension. That's basically what this does in one line. So that's a very typical part. Oftentimes we need to do that. So once we have this 2D matrix, we can do our custom analysis, whether it's a GLM, regression, correlation, or some kind of machine learning. So you do whatever you plan to do. So that's the image reshape. That's a critical part. Actually, here these are the voxels. As we know, this is something like that's this number from. Most of them, about at least 2/3 of them, are zeros after pre-processing. Why? Because it's outside the brain. So those are all zeros. So actually, only about 1/3 of this image, these voxels are meaningful. So to speed up the thing, we often use some kind of mask. We do that analysis only on those 1/3, so we can save some time. But for today's example, I'm going to show you, I don't care about it because it takes less than 1 second. So next one, I'm going to mention something about our analysis. That's often a time we need to do our special analysis. Or otherwise, the software packages like FSL, AFNI can do pretty much what we want. So our analysis we often need to do by ourselves. So sometimes we need to define our own ROI, and sometimes we use a predefined ROI by literature or by other researcher. They give us, that's what we use. The good things, it's kind of independent of our own analysis. We don't get the you are double dipping. You use your activation to define your own ROI. That's often not optimal. So when we use ROIs, we need to pay special attention. The ROI we use should be the same NIfTI form as our functional data. That's, remember, same form. Otherwise, we don't know what we are doing. So that's why here I'm going to show you one of the example. NIfTI view. It will start with a 2mm template. Let me get a better one. 1mm. Looks better. Okay. This is, as we can see, is MNI space. So if we want to use the ROI, for example, I have something. Let me add overlay for. For example, let me load a AAL template. You can see this is a anatomically labeled automatic anatomical labeling, AAL. Label some brain structure based on anatomical structure, anatomical feature. So if I turn on off the AAL like this, you can see they overlap pretty well because they are all in the same MNI coordinate. So if they are not in the same coordinate, you will see a mismatch. When we use ROI, because this region of interest is defined in MNI space, we should use our functional data in MNI space. What happens if not? If not, we need to do a transformation. Either we transform our functional data into MNI, then we can use, this blue region. We extract time series from this blue region from our data. Or the other way, that's often simpler. We transform the MNI ROI definition file into our own space, same space, our functional data. Either way, they should be equivalent, but the latter should be easy because this file has it's very small, maybe it's only 1K. Our functional data may be 400 megabytes. So, but either way, the other advantage of what to transfer our own functional data is whenever we transform our functional data, it involves interpolation. So we try to avoid interpolation if possible.
Hey, Xiangrui, we have a question from the chat. This is coming from Juan. So, hi, is there a specific data type / object for 4-dimensional voxels? Say if you're working with a video and need to represent the time or frame position.
Is there a specific data type for 4-dimensional voxels? Oh, actually, what we are talking about is 4-dimensional. Like here, 1, 2, 3, that's space, that's time. But in our case, the time resolution is 1, each time interval is 1 TR. In this case, 2 seconds, pretty slow. But in the movie, it's like a video, like a move MP4 video, that's typically like a 30 frames per second. So that's but in that case, it's not 3D. Movie is only 2D. I think 3D. The third dimension is a 3, RGB color. It's a 3, but that's a X, Y, Z color time. So typically people want saved video as NIfTI, although you could. So, but NIfTI is mainly designed for brain image analysis, not for the video analysis. Video, you have a lot of, I think the industry has a lot of tools for video part. It's because a high frame rate, it's a lot file is often very large. Okay, make sure the image data and ROI definition use the same form. If not, we need to do a transform. The transform may need a transformation matrix between the two forms. For example, typically our data is in the scanner anatomy space, and oftentimes ROI file is in MNI space. Okay, here I'm going to show you an example from the image reshape all the way to image analysis, image visualization. So these slides I have mentioned in the first seminar, but we didn't go into detail. So this is a general linear model, and Y is our image data, like I showed it to you here. Y, that's our Y. Could be a 4D, we reshape into 2D. And we can simply treat it as 1D because all these voxels are independent. We can pick any of voxels, only treat at y as one dimension with 138 time points. So the X is the design matrix. That depends on how we run the experiment. For the example I'm going to show here, let me see, what did I load? I load resting state. Okay, let me load another one. Let's load another one. The task one, yeah. Then we do the same thing. What's that? Oh, look there. So now we have the Y. Oh, let's find the previous line. Yeah, we have a transpose. So we want this time points voxels. In this case, 182 time points. Okay, so X, I think I have the design for the X. So yeah, I think this is a text file, although it shows as a .mat file. That's from FSL. Yeah, it is a text file. That's a FSL design matrix, some header and 2 columns. And I'm going to copy, paste these 2 columns assigned to X. Let's say Y and X. So Y is our image data. X is 182 by 2. That's the 2 columns. Let's take a look. What does X look like? Those are the 2 columns of X, one blue, one red. Those are the different 2 trial types for the X. So I'm going to tell you what are those 2 types later, but this one, so we already based on the onset time, we come up with HRF function to construct the design matrix. And this actually I directly export from FSL. So if we check the mean value of X, they should be 0. Yeah, it's something times e minus e power to 10 minus 9 is basically it's 0. So the mean is 0. Is remove the mean. That's a very important step for the design matrix. Okay, now the beta is the effect we are going to get. One beta for each column of X. And pay attention that beta is irrelevant to, the dimension of beta is irrelevant to the number of points in Y, because the X should be having exactly the same number of length of Y. So this is the same as the example I gave you for the general linear model. The number of TR here is the number of equation. Number of predictors is the number of variables we need to explain. That's the number of column in Y. So in Matlab, here is if we organize the Y in that time by voxel, that's the thing we do in Matlab to solve the linear equation. It takes about 1 second. So let's take a look at what a B. B is, oh, actually that's not right. Okay. Y is not right. So we need to, because if we plot some Y, let me plot a red voxel. Huh. Unfortunately, I get someone outside the brain. So I don't know which one's inside the brain. Huh. I'm so unlucky. Whew. Because 2/3 of them are 0. This is outside the brain. Hey, I can't believe. Let's plot all of them just. That's too slow. Probably it's going to crash. So what the point I want to make is the mean value of the y, this is from FSO, is about, for those valid voxels, it's about 10,000. So that's the that's because FSO did that way purposely. So we need to do this before we do this. We need to get rid of the mean. After this, the mean y is 0 for all voxels. Note that this only works for Matlab 2016B and later. In early one, you will get an error for this. It won't work this way. So now we do this. Okay, so the dimension B should be the same, like this. As 2 variables, this is the number of voxels. So so this one, by minimize the variance E, let's compute the E. The error term equals data minus design matrix times our solution. So in the ideal case, like the 2 variable, 2 equations, we are sure it explains data perfectly. That means the error term or 0. But in this case, this has the same dimension as y. So let's take a look. Y, X, B, E. So E has the exact dimension, same dimension as Y. X our design matrix, B is our solution. Two values for each voxel. So oftentimes we are interested in the T map. For example, this example, what we are going to do here is we want to say, this will give an error. So we will want to define C as first one versus the second one. So one minus the other minus one. That means for these 2 beta, we will do a sub difference between the first and the second. That's what typically in our analysis is called a contrast. So then we do the T. Look, this takes less a second. The T should be in the dimension of this, 1 by this. Yeah. So basically, it's the difference between first and the second for each voxel. The voxel they are always independent. So now let's quickly create a result. I'm going to call it result. Just I'm going to simply same as NI, but I'm going to reduce the image to only to 1 dimension, 1 volume. X, Y, Z, and the last dimension is the same as just only one volume. So now the trick for this one we are going to store all our t values into this NIfTI image. That's the trick to keep this dimension, but only assign the corresponding t value into each volume. Now we can do update by the NIfTI tool because we changed its dimension tab. So this will fix the header. So now, where's the NIfTI tool? Oh, okay, I'm going to show you something weird. Turn this off, add this. We will see something weird. Haha. That's our activation. That doesn't look right. Why is that? Different NIfTI format. So, because this MNI, this is our original space. So that's not, the overlay doesn't make any sense. So I'm going to open the original NIfTI, the MP range. So we can do a meaningful overlay. This is bring for that particular participant. Now I'm going to do the result in the memory. I even didn't save files. We could do a NIfTI save. That looks inside the brain, right? So we can do an interpolation and two-sided to increase the threshold. Something here. Okay, now we have the activation. So for this particular task, that's the contrast between the two conditions. Actually, this is a theory of mind task, one called false photo, the other called false beliefs. So those are the contrast, the activation. They look quite similar to default mode network, right? So that's if you check the theory of mind activation, that's pretty much the case. That's why I show you how easy it is to do the to do a general linear model in Matlab. Use the NIfTI tool all including my NIfTI tool package. So let's remove this. This is ugly. This doesn't make any sense. Okay, here I'm going to show you the ROI. I think that's all for the PowerPoint. Yeah, I think so. Yeah, that's all for the PowerPoint. So for the ROI analysis, I keep seeing we should have the same form. So what happens if we don't have the same form? For example, this NIfTI file, for example, we want to use this region. Let's see, this region to extract the time course for this data set. But we know this data set is called S form. S1. One means anatomical, like this one. Scanner and scanner coordinate. This one is MNI, the standard space. They are not same, so we can't directly, even if they have the same image dimension, those dimensions won't match in space. So if we use that, that's ridiculous. So how do we do that? So earlier I mentioned we can transform either one into the other space. So one way is, for example, this is the subject brain. If I, where's the, if I overlay this one to the scanner and anatomy, you will see a warning saying the transformation is inconsistent. So probably the the overlay is meaningless, you can say, they still overlap a lot. That's because MNI use ACPC as a origin coordinate. For the scanner space, we try our best to do that when we set the participant in the scanner, but each participant is different. It's not accurate. But it's not completely off, but we can see it's off by something like 2 centimeters. Not there are plenty of overlap. So that's what we don't. Let's get rid of that. So the way in my tools, there's another overlay. You can add a line overlay. In this way, we will add the AAL into this one, but we do need a transformation matrix to transform the MNI AAL NIfTI into our scanner space. So typically we don't have that. But in the FSL, when we do the analysis, we have the other one, like this guy, is we use that to transfer the functional image into MNI space. No, not this one. Yeah, this one. Oops, this is some retrieval to standard. Yeah, this one. Let's see. 16 numbers. That looks familiar, right? Yeah, it is a transformation matrix. In this case, this 9, 3 by 3 is a rotation part, like the transformation matrix for NIfTI. Those 3 are translation. So the diagonal major I mentioned is 1, 1, 1. They should be very close to 1. The other 9 numbers, the other 6 numbers, they are all close to 0. That means diagonal. So if it's not diagonal, typically the Dicom NIfTI converter make the diagonal major. So in this case, not exactly 1, because when we do the alignment, this brain to align this brain to the standard brain, there's a resizing part because the brain size doesn't match. So a little bit, like 15%, 10%, 17%. That's from this guy to this guy. But here we want to transform this guy into the, here, that's opposite. So that's the opposite. In Matlab, it's so easy. So I'm going to load this file into Matlab. That's those 16 numbers. So the opposite is simply invert of R. Still, this is 0, 0, 1, but the other is the inversion of the matrix. So if we save this 16 number as this, we name that as standard 2 retrieval. That's the opposite. I already saved a copy of that. So we are going to use that file. And I'm going to pick the AAL. Then I'll ask for the transformation matrix, FSL transformation matrix. Where's that? It's a standard to this one. That's that contains these 16 numbers. Oh, I guess I chose the wrong one, right? Because it doesn't look right. Oh, no. The reason is I'm opening this MP rate. What I want to open is the functional image, because this transformation matrix is from the functional image to the standard, not the MP rate to standard. Look how easy it is to make the mistake. Let's open our functional data. That's filtered function. This guy. This is the 4D data. This doesn't look beautiful because that's a 3 by 3 by 3 million resolution. The number of volume is 182. We already saw that. So now we overlay the AAL. Here's the AAL. Oh, sorry. I forget use the wrong one. Use this one. Then it asks for the transformation matrix. Yeah, that's the one. Okay, let's remove the wrong one. Look, now they match pretty well. That's because we transform back into the our image space. In this case, you can save this as an extra version, say another NIfTI file that will be in the image in our functional data space, image space. Then they have the same number of voxels and the same orientation, same transformation matrix. They have a voxel to voxel correspondence. That's the way we should be very careful to use how we use the correct region of interest. For example, if we want to use this brain region, that's how we use it. So lastly, let me show you a beautiful picture for the 3D, for the surface-based HCP data. For example, for here. So turn off this one. Because it's surface-based, you can see some only part of voxel are used. But here we have a lot of those region of interest. Like a different interest of, if I click here, it shows in the 3D space is over here. So that's the HCP data format. So you could easily visualize this on the surface space. So this visualization tool should be very convenient for you to use. Yeah, I think that's the NIfTI part I'm trying to emphasize. So the only very important thing is that people very easy to make a mistake is the ROI. So when you use ROI, it's left, right handedness. So if you see a transformation like this, positive, 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 that's right-handed. That's the standard one. But for historical reason, if you open any Matlab, any FSL file, you will see this number is negative. That's left storage. This number negative, it doesn't matter, positive, negative, they can be same data set. But in this case, it's simply first dimension is flipped if this is a negative number. So if you see two transformation metrics, they are not the same, be very cautious. Because not only the positive, negative, because image are flipped for the first dimension. So I think that's mainly for FSL. For other packages, pretty much they all use right-handed. Okay, any questions? You can unmute and ask, or it seems we have it, or type in the chat.
Thanks, Xiangrui.
Yeah. If there's no question, I will thank you very much for coming to the seminar.
Thank you so much, Xiangrui. This is
Yeah,
great.
you're welcome.